So my lab has developed Hydra as a model for developmental biology, but also for host microbe interactions in the last 2 decades. And I just briefly summarize here our major interests and some of the recent findings. I just remind you that this model is maybe one of the most oldest models in biology. It was invented 1744. I'm asking where C. elegans and the mouse and the fly were at that time. Margaret McFall and I, we just thought a little bit about that in the last few months, and we put out 2 papers which compare model systems in symbiosis and model systems in developmental biology, and they kind of overlap, but not totally. And we just stress the importance of model systems to understand the complexity behind development and symbiosis. If we turn to Hydra, this is a classical developmental biology model for many, many years, and it's worked out the complete cell biology, most of the molecular biology, the genome, many transcriptomes, etc. And we know pretty much the pattern formation mechanisms which are going on during a normal asexual life state. We also discovered many years ago that in addition to the cells, the Hydra cells, there is, as in any other organism, there is a stable microbiome living in the glycocalyx, a kind of a mucosa outside the ectodermal epithelial cells, and there is a strong interaction between both of these systems. So at Kiel University and in a collaborative research center with Hinrich yesterday introduced to you, origin and function of meta-organisms, we are trying to understand the interaction in Hydra, but also in many other systems at a functional level. It's very clear and very general, and all of you know that quite well, that we start to get a real good understanding of the importance of the microbiome in development, in the immune system. We get evidence that it's important for neurogenesis. It also is important for growth and for maturation of many organ systems. In Hydra, by establishing a germ-free Hydra culture, which we now have for several years, thanks to a postdoc in the lab, Jinru He, we start to uncover amazing difficulties of the animals in the absence of microbes to do their normal development. And so this is just at the end of, in a pre-publication phase, but the animals just stop to asexual proliferate, and they make bizarre pattern formation problems. And thanks to the old history of Hydra and all what we know now numbers of gene families, signal transduction cascades, receptors, transcription factors, which all are affected by the absence of the microbes, and we can study that interactions now very much in detail. And just in the last few years, my lab showed that FOXO, which is a critical stem cell transcription factor, is a crucial mediator between tissue homeostasis and the microbiome. The Wnt signaling is influenced by microbial colonization. In the absence or disturbance of a microbiome, the tissue develops tumors, and I come back to that in 1 minute. And I would want to spend a few minutes on our recent interest in understanding the neuron-microbe interactions. Coming back to the tumor paper, this was an amazing story. We knew that there is a tumor. We have published that in Nature Communications some years ago. But only recently in that paper here, we could show that the tumor actually is caused by a dysbalance, by a dysbiosis in the stable microbiome and an environmental microbe, which is per se harmless, a spirochete, in the combination with a beneficial microbe, a Pseudomonas species, causes the tumor. Nervous system. These animals have invented the nervous system, and the nerve cells are in direct contact with the area where the microbes live. And so we hypothesized that this very ancient nervous system is in contact with the microbiome. And actually, we have proposed that the nervous system only exists because and with the microbes. You cannot separate that from each other, and I will show you evidence for that. In an earlier paper, we could show that neurons produce neuropeptides, which are antimicrobial peptides and which affect the microbiome. And we genetically knocked out this neuropeptide, and then we could drastically disturb the microbiome, both in its composition and in its spatial distribution along the body column. We next then asked, do the microbes do something with the neurons? And the answer is yes. And so we went to a simple reflex or behavior, which is very ubiquitous in the animal kingdom, spontaneous contractions. That's doing your gut at the moment. This is your small intestine. The worms do it, the corals do it, the jellyfish does it, Hydra does it. And if you look into that in more careful, we could show that in the absence of microbes, this spontaneous contraction is drastically reduced. And if we add back the microbes, but only in the normal composition, we can nearly rescue that effect. So that means that there's a complex microbiome. It needs diversity. This produces something, and this something activates a neuronal receptor. And the question is, how does that really work? And so the molecular architecture of the nervous system could only be achieved with a fantastic work of postdoc Alexander Klimovich and modern single-cell transcriptomic technology. And we first could show that very conserved from human sequences ion channels are the feature of the ancient pacemaker cells, which are responsible for that contractions. And they are about 20 cells, which are in the critical body area, which are controlling these contractions. And when we inhibit that, then the body contractions go down or get drastically reduced. So there is a functional evidence for that. The microbes indeed influence these contractions. And that is also, of course, conserved in evolution and also in your gut. And there's a modern disease, obstipation, actually also related to Parkinson, where this slowing down of the contractions and getting irregular is causing pain. And we are working on the mechanistic details, how that really works together with the microbiome. The single-cell transcriptomic profiling showed us that there are 7 subpopulations of neurons present, and they all are characterized by their neuronal specific genes and transcription factors and neuropeptides, but 70% of the neuronal transcriptome are immune genes. So these are TLR, so toll-like receptor pathways, C-type lectins, NACH domains, antimicrobial peptides, etc. And we were very much surprised. Why do neurons express so many immune-related genes? And the answer is getting very clear. And if you think back of our hypothesis, the neurons use that repertoire of immune cells to manage the beneficial microbiome, which is in close contact to them. In addition to the conserved genes, which you can find by conventional database analysis, we could show that a large proportion of the transcriptome of neurons is made up of non-conserved genes, which we relate to as taxonomically restricted genes. You may find them in the literature as orphan genes or non-conserved genes. These are very abundant. People ignore them because they are hard to work on. You cannot just look into a paper and predict what they are going to do. So you have to invent functional assays. We did that by machine learning programs, and we could show that these taxonomically genes really are also important in controlling the beneficial microbiome. I just mentioned that because in all of your data, in all of your organisms, you will find these non-conserved genes. Please do not ignore them. They are more than just orphans, as we put in already more than 15 years ago, nearly now, in transient genetics. They are important in evolution. So summing up and coming to an end, it's very clear that the nervous system evolved in the presence of a constant microbiome, and that has had dramatic impact on the anatomy, the morphology, and the molecular architecture of the nervous system. There are conserved ion channels which are talking to the microbiome. These ion channels are in the pacemaker cells. They go to muscles and control the body contractions. But there are also conserved immune genes which take care of the microbiome, that it is in a stable situation. And when you have genetic problem, or when you have an environmental problem, that the microbiome is changed, you develop, your body is in real trouble and develops diseases. This is, as you have shown in that paper here, this is a very conserved process. You find very similar processes if you directly compare Hydra and the mouse, or we also included in this paper, the C. elegans. So that is actually what we are going now in more detail. I'm very much interested in this neuromicrobe interactions. It's very clear that from the evolutionary beginning, neurons got signals from microbes, and microbes talked to the neurons, and the neurons secreted neuropeptides, antimicrobial peptides, to keep the whole menagerie in a balance, and together that forms the holobiont or the meta-organism. With that, I'd like to thank a wonderful group of people, and I mostly talked about the work from postdoc Alexander Klimovich. He is supported by graduate student Christoph Gietz, and currently also by postdoc Jinru He, who develops the developmental model of Hydra. And the work is done in the Collaborative Research Center, Meta-Organisms at Kiel University. I thank you very much for your attention, and if you have questions, feel free to address them.
Thank you very much, Thomas. There is one question from German. Go ahead.
Hi, again, it's actually two questions. Thanks. It's a beautiful system. The first one is, do you know how generalizable is this model system to other cnidarias or to other hydrozoas? And the second one is, how specific is the response? Like, does it depend on specific microbes, or it's just generally just having a microbial community would elicit these responses? Thank you.
Thank you very much. Very good question. Hydra is, of course, a member of a large animal phylum, the cnidarians, and there are many, many different groups, and all of them are kind of studied in various laboratories and at the various contexts from an ecological point of view and others. I'm brave to say that none of these the other models as developed as Hydra, and you cannot do we can do genetics in all kind of ways. We are and very tricky. So visualizing the cells, making them transgenics, knocking out genes, overexpressing genes. We have a genetic ablation system now working where you can deliberately ablate certain subpopulations in the tissue without disturbing the tissue. There is no other cnidarian system which is similar to that. But I'm very happy, and I'm frequently talking to coral people, for example, to support them to develop that in that direction. And if there's anybody in this SymbNET group here, we are very happy to support any of these efforts. The second question is also a very good one. And the microbiome in this model system is very specific. It is species specific, and that was a finding which triggered that from the very beginning. Every single species has its own microbiome, and it seems to be co-evolved because we have a very nice phylogeny of the Hydra species, and we know the microbiome very well. And so the most ancient animals have a very different microbiome from the more recent species, etc., etc. So it's very specific. And the question is, how do you maintain the specificity? That's not, I mean, we have species-specific antimicrobial peptides, etc. We keep the animals never go out in the nature. We keep them in plastic dishes, and they maintain for decades in the artificial environment their specific microbiome. Pretty amazing, but wonderful to study.
Thank you. There's another question from Gonçalo.
Yeah, thank you, Eli. Thank you for the talk, Thomas. I was actually going to ask about the specificity and how it compares to the environment of the Hydra, but you just answered.
Thank you.
So, yeah, never mind. Thank you.
Great. So if there are no other, oh no, there's one. Nelson, go ahead. Yes.
Hello, Thomas. Very nice talk.
Thank you.
I don't know if I missed it, but you told us that the neurological cells can communicate through these antimicrobial peptides, right?
Yes.
With bacteria.
Yes.
And the other way around, how do you think it happens?
By small, very good question. These are neuropeptides. So first of all, the one way is by neuropeptides, and but remember, we also discovered novel genes, orphan genes, and we had to go a hard way to identify with machine learning which could be antimicrobial peptides, and then we tested them functional and we found them. So the general way of the neurons to the microbes seem to be antimicrobial peptides, and the way of the microbes to the neurons seem to be, and we are working on that, it's not published yet, small molecules, very small molecules, microbial metabolites. I'm collaborating with a wonderful colleague in Munich on mass spec analysis. And so it seems small molecules which somehow trigger the receptors, the neuroreceptors.
Okay, thank you.
There's one more question from Sudeep.
Hi, hello. It's fantastic work. A very interesting system. I was actually kind of a similar question as to you said that the microbiome is recognized by the Hydra and then it produces antimicrobial peptides. And I'm just trying to draw parallel with the higher organisms. Is there anything like cytokine production which may be happening in the Hydra and such molecules which are communicating with each other?
Yeah, well, it's interesting. You're walking while you're talking, and in the same time you're taking part in a meeting in Portugal. That's pretty amazing. So the question is, the cytokines seem to be very specific for the more advanced animals. But I think the comparison, what I would advise you is looking for the small molecules. Short chain fatty acids are kind of a whole black box of molecules which are produced by microbes. And so in this category of small metabolites, I think is the communication which is conserved from the very ancient organisms up to humans. And so that's where we are looking into.
Okay, thank you.
Thank you, Subit.